So this is the last segment on humoral immunity, and of course you couldn't have humoral immunity without your B cells or your B lymphocytes. So this segment is devoted to understanding, or just outlining rather, where the B lymphocytes come from, how they mature, and then lastly, how they respond to antigens and are activated. First off, millions of B cells mature in the bone marrow. This is a statement I've already made previously, and you should note the relationship between the B there, B cells from bone marrow and T cells from thymus. So what I've done here is I've drawn just in a very cursory kind of way a number of B cells. You can tell because they've got B on them, I hope. And then rather than drawing a whole halo of receptors around them with this kind of unique antibody binding fragment on their outer surface, I just drew like one with this one exception here. I got a little crazy and drew 4. But anyway, so what happens, you have millions of these B cells, these B lymphocytes maturing in your bone marrow. As illustrated in my simple cartoon over there, each cell is covered by a single type of unique receptor. And you know from our previous lecture that these are going to be IgDs and IgMs. And of course, what makes them unique is going to be the antigen binding fragment, the FAB. And I've illustrated that in terms of using different colors now instead of shapes. So we have red and various shades of blue and purple and green. And you see that each one of these B cells has associated with it one receptor. Of course, it'll be surrounded by hundreds of receptors, but I just have one illustrating the uniqueness of each of these B cells that's produced by the bone marrow in terms of their having a unique FAB or antigen binding fragment. So you have the potential here now to generate thousands, if not millions, of different types of antibodies if you just activate one of these cells, and we'll talk about that. Now we have a problem in that you don't want to generate antibodies against parts of the fetus. And so one of the first things you have to do in terms of survival is to kill all the cells that would produce antibodies against you. And so that's what happens. So clones with receptors that are directed against fetal antigens, or what we would call self, are killed in fetal development. Obviously, you don't want any form of autoimmunity to develop at any time, and so then the body will eliminate certain clones that would potentially produce antibodies against your major histocompatibility complexes are the things that you might find that are unique to you in your blood and tissues. All right, so that's to do with the production and formation. All right, now in terms of activation, there are two types of activation. One that's mediated by T cells and one that's not. I looked at the one that's not mediated by T cells. It's very short-lived. It's also very weak, and so I figured it was very unimportant. So don't worry about that. So just worry about the activation of B cells by T cells. And so in this case, we're looking at T cell dependent activation. Of the B cells. All right, the first thing that happens is that, of all of these different receptors, all of a sudden now we have a challenge where there's an antigen in the blood that's specifically recognized by the one I took the most time to draw, obviously, this one right here. All right, so here we have our antigen. And this antigen now is very specifically binding these receptors and only these receptors on this particular cell, and not any of the other B cells that we just erased because we're not interested in them. All right, so once a B cell binds an antigen, and of course the only way antigens are going to get in the blood is if something bad happens. So all of a sudden, something bad has happened. We have this antigen. We have to deal with it. So how do we recognize it specifically? Well, of all of the different clones of B cells, there's hopefully one or two or three that have receptors, these IgDs and IgMs, sticking out there that will stick to the antigen. All right, so the antigen and these antibodies, these immunoglobulins that decorate the B cells now are stuck together. All right, so that's the first thing that happens. Is the antigen binds to a specific naive B cell. That has a complementary FAB. All right, now, basically after this, nothing happens without the intervention of a T cell. So this is the first thing that happens, is the antigen binds. The second thing that has to happen is that the B cell needs some help from a, T helper cell. All right, so once your B cell is activated, it'll attract the attention of a T helper lymphocyte. So the T helper recognizes that the B cell's got a problem here. They form a relationship. That's its job. Help. Okay, so B cell presents this bound antigen to the T helper cell, as illustrated there. All right, and then the T helper cell secretes a chemical that works kind of like a local hormone or a paracrine called cytokine. We mentioned cytokines previously as used in cell-cell communication. So this is a very specific type of cell-cell communication. So the cytokine will look like this. All right, so we bind the antigen. Let me label that. To the B cell presents the antigen to the T helper cell. All right, and then 3, the T helper cell is secreting these cytokines. And these cytokines then are going to activate the B cell. All right, these little green stars that I'm drawing represent cytokines. So these are small molecules that'll be secreted by the T helper cell. All right, and these cytokines then so continuing on with 3, maybe this should be 4. So the cytokines then are going to cause a clonal proliferation of this B cell. So it's going to double, make 2, double again, make 4, double again, make 8, 16, 32, 64, 128, 256, 512, 1024, 2048, and so on and so forth. So you see it's exponential. It's going to double and make thousands of B cells exactly like itself. All right, so the cytokines cause a clonal proliferation of the B cell. Not every B cell of this specific B cell in which this T cell has a relationship. So what happens then is that now we have more B cells of this exact type. That's why we call them clones. And more B cells of this exact type, again, clones. And you know, these cells can double every 10 to 12 hours. And so, you know, you can easily go from one cell to thousands of cells very quickly with this doubling. And we're going in two different directions here. So in one direction, we're going to make what's called plasma cells. All right, and plasma cells have the important task of mediating this humoral immunity. They have the important task of making the antibodies. All right, so that's one direction this clonal proliferation is going to take. And of course then these antibodies will be very specifically directed against this antigen, because it was this antigen that caused the trouble in the first place and stimulated this particular B cell to make antibodies against it. All right, so that's what all of its progeny are doing on this side, is they're all plasma cells, they're all making antibodies. The antibodies all look like the antibodies that are on the surface of that B cell. That kind of makes sense. And so that you can visually now see the connection between the receptors and that were originally used to detect the antigen and the antibodies that are finally being made here by the plasma cells. You see that they are identical in nature in terms of their FABs. All right, meaning that these antibodies specifically recognize this epitope on this antigen and no other. Now the other way is, so in this is again the specificity side of this acquired immunity. Remember, the other side of this acquired immunity is memory, and so that's what we're going over here. These clones on this side, some of them are going to form memory cells. And memory cells are going to become quiescent or quiet. They're not going to do much until they encounter the antigen again. All right, so if the memory cells encounter the antigen again, and not just any antigen, but this very same antigen that started out everything right here, if they see this antigen again, then they're able to mediate a rapid, robust secondary response in terms of antibody turning into plasma cells and making antibodies. All right, so if the memory cells encounter the same antigen, they will make plasma cells, again, like here, and the plasma cells will make antibodies fairly quickly, and they'll mediate this very rapid, robust response that you know as the secondary response. Okay, so that's everything you need to know about, at least what I think you need to know about humoral immunity.